of above and below ground fluxes, and this is being presented by Dr. Noshin Ilyas, Associate Professor, Department of Water. Over to Dr. Noshin.
Thank you, sir. Bismillahirrahmanirrahim. Assalamu alaikum. First of all, I am thankful to the Director Advanced Studies for providing me an opportunity to discuss some of the work which is being done in my lab. I hope I am audible and you are able to see my screen. Sir, my screen nazar aa rahi hai?
Yes, madam. Fine.
It's fine. Okay. Thank you. So I welcome you all. And the topic of today's discussion is the regulation of above and below ground fluxes for combating the climatic changes. As all of you would be knowing, I'm Dr. Noshin Ilyas, and I am working as an associate professor in the Department of Fertility at Agriculture University, Rawalpindi. So why we are going to discuss about all the fluxes and why they are important. Some brief thing about agriculture. I'm not going in detail of these things. You know that we are an agriculture-based country, and this agriculture is contributing to 22.7% of our GDP. Along with it, we have a large area which is under the cultivation, and we are the top producer of many cereal crops like wheat, cotton, sugar, along with some other plants like mango dates and rice, cotton, and sugarcane. They are considered as our major cereal crops. And we also have some minor crops, but these are the major cereal crops, and our whole dependence is upon these crops. There are some things which are important for the agriculture in Pakistan. The role of agriculture, as I just defined, that our economy is dependent upon this agriculture. And this agriculture is a basic source for our food, our fiber, for our industry, for our foreign exchange, and is also for a market for industrial goods. Now, there are some things which are our strength, and there are some things which can be our weakness. Among the strength, we are a very diverse country. Along with it, we have a large area which is under the cultivation. We have one of the largest, the network for the irrigation of the crops. But there are some threats. One of the major threat is that we have a great population burst. We are the 6th most populous country, so we have a huge population to feed. Along with it, we have some serious food security issues. And when we are talking about the diversity, it also means that we have a great diversity in our geography. And this geography diversity, it also makes us vulnerable to various climatic anomalies and the disasters. So these are some of the threats which we are going to face and which we are rather facing nowadays. Climate change is no more a threat for the future. It's happening right now, right at the moment with us. And the first thing which I want to address over here is that we are having a global hunger index. We have a severity for global hunger index. It means that we are having a severity of 24.7. It means that this amount of population, this is not having the amount of the food which they require. Along with it, when we are talking about the production of the food, water is the basic necessity for the production of the food, but we are also considered as a waterless country. Our country has very limited amount of the water. We are not among the countries which have less water. We are among the countries which are having rather no water. And this condition is also very severe. We are having 2.56% of water only. The most important thing is that we are the 7th most vulnerable country to the climatic change. We are vulnerable to all the climatic disasters, the anomalies, the abrupt weather changes, and they are all impacting upon our agriculture. And due to this, the serious threat is that our agriculture is predicted to have a decline rather than increase, although we need more food for our growing population, but the predictions are that we are going to have a limitation or negative or decline trend in the production of the food. So this impact, it is indicating to be up to 25% decrease, and all of this is a very alarming situation in our country. The other important thing when we are talking about the production of the plants and particularly the crops is our soil fertility status. We do not have any organic farming, only as per the International Organic Farming Association, our organic farming is less than even 0.1%. Along with it, the most important issue is that our soils are reported to be nutrient deficient, and the soil carbon is less than 0.52%. We are also deficient in most of the nutrients like nitrogen, phosphorus. There are a number of reasons for the low soil fertility. the parent rock material, the soil erosions, the less nitrogen fixations. Along with it, if you can look on the right side of the picture, there are some data that is available. That is the fertilizer supply demand situation. Our fertilizer demand is more and the production is less. So we are having a shortage over there also, which also results in the increase in fertilizer hike. I hope you remember that this hike was very much reported in the last couple of months. Along with it, there have been numerous data that has been published that our fertilizer use efficiency is also low. It means that we provide more fertilizer and we get less productivity. Now, what is the need of the hour? Need of the hour is that we have to define what are the major climatic stresses which are going to happen in Pakistan. Like we have the issues of the drought, we have the facing salinity, the temperatures, the pollution is also very increasing at a very alarming rate in Pakistan. And how we can combat these climatic changes? We have our main focus is the plant. And when we are talking about the plant, there are two basic components. One is the one which is present above the ground, and the other one is the one which is present below the ground. So we have to target both the fluxes which are happening at above the ground and below the ground. When we are talking about the above ground fluxes, we need to understand the abiotic stress tolerance mechanism in the plant, the changes in the morphology, the physiological responses, the biochemical profiling, and molecular mechanism. We have to understand how a plant is able to combat or how a plant is able to withstand ameliorate the stress conditions. Along with it, equally important are the below ground fluxes. Among these, we have there are very important group of microorganisms which are considered as the plant growth promoting bacteria, which have the ability to promote the growth of the plant. So they are targeted for the improvement of the plant, particularly for the stress mitigation. Then we have some soil amendments. We need to study about the changes which are happening in the root and also on the soil microbial diversity. So what are the contributions by me and by my lab in all this? Our work, this is, we are basically have defined our work into three parts. In the first part, we are studying into any kind of the stress, either it's the drought, it's the salinity, or any kind of the stress. We are targeting the plant stress tolerance mechanisms. Then we are targeting the micro which have the ability to tolerate the stress and also mitigate the stress when inoculated. We are also studying the role of the various soil amendments in improvement of plant responses to the abiotic stresses. Then we are combining all these three because the target is the plant responses. The ultimate is that we need to transfer these responses into the field and we have to improve the plant performance in their native environment. So this is the research work, which is, I have just mentioned, we have divided into three parts. The first part is the study of the plants for the stress tolerance mechanism. In the second part, we isolate the indigenous plant growth promoting bacteria from the stress ecosystem. The idea is that if we have the ability to isolate the microbes from an area which is naturally under the stress, they would naturally have the ability that they are going to perform better under the stress condition. Then we characterize these PGPRs, we go for morphological, physiological, and molecular characterization. I hope you know until we do not have the exact identification, we cannot use these microbes for later studies for the inoculation. Then we have studied a number of soil amendments. We have worked with till yet, we have worked with the compost, with the biochar, with the nanoparticle, and some alternate cropping techniques. Then we combine these two and we study the effect of the microbial strains and soil amendments on the plant morphological, physiological, and biochemical parameters, particularly under the stress conditions. We also have tried in some parameters to go for the molecular mechanisms to understand what's happening inside the plants. So our main target is two things. One is the plant growth promoting bacteria, and the other are the soil amendments. Plant growth promoting bacteria, plant associated bacteria, and they confer the plant growth advantages to the plant host and through the nutrient uptake, through the hormone homeostasis, and they're improving the tolerance to the abiotic stress factor, and also along the biotic, including the phytopathogen. Similarly, soil amendment is any material which is added to the soil, and it improves the soil physical and chemical properties. It improves the properties particularly related to the plant growth, like the water retention, the permeability, the water infiltration, the drainage, aeration, and structure. The ultimate goal for the soil amendment is the improvement or the better environment for the roots of the plants and for the microbes. These are the stresses which we have targeted up till yet. We have studied the drought, salinity, heavy metal stress, and hydrocarbon pollution in our lab. And see, these are some of the examples of the studies which we have carried in the lab so that we can reach to something which can go into the field. The first study, the first test which we studied that was upon the drought tolerance. What we did was in the first part, we studied the stress tolerance mechanism in the plant. How this plant is going to, we targeted the main crop, that is the wheat, that's the staple crop of the Pakistan. We studied that how this crop is affected by the stress and how this plant, the wheat plant, it combats or develops resistance against the stress. How it's this drought stress, it alters the crop phenology, the changes in the root shoot ratio. Most importantly, we studied the targeted the plant physiological mechanism, including the plant water potential levels, the osmolyte productions, the production of the reactive oxygen species and the antioxidants. And along with it, in the end, we did some QTL mapping of the important traits. In this QTL mapping, we tried to find out that these important things, the important parameters which determine the resistance or the tolerance in the plant, which differentiate a plant from the being stress tolerant or being stress sensitive, we try to find the location on a particular chromosome in the wheat genome. And this is for all the ABD genomes for the 7 chromosomes. We did these studies and we did the QTL analysis, and this work was published. Later on in the second part, we did was the isolation. We isolated some of the plant growth promoting rhizobacteria from the soil, which had been collected from the stressed regions, which are naturally facing the water shortage. Then we screened these microbes based upon their tolerance, particularly to the different water potential. Then we screened these microbes for various plant growth promoting traits. These plant growth promoting traits include the indole acetic acid, the hormones, the indole acetic acid, the gibberellic acid. Along with the ACCD aminase is also considered a kind of harmonic activity. Then we did for some nutrient production, like we did for the iron solubilization, the phosphorus solubilization, and the siderophore production. The best traits which we selected, they were later on characterized for the phenotypic and the genotypic characterization. We did the 16S RNA sequence analysis, and this 16S RNA sequence was carried on for the blast analysis, and then the gene analysis that was obtained, that was submitted in the NCBI, and the accession numbers were obtained. These are some of the promising strains which we have obtained in our lab, and these were, the first one is the name of the strain. We have some good strain of the Bacillus, the Azospirillum, the Rhizobium, and these had been isolated from the arid regions of the Pakistan, from the salt regions of the Pakistan, and from the number of the hosts like the wheat maize and the salt grasses, and these are their accession numbers, and these are the production of the stress hormone, particularly the abscisic acid, which was tested under the conditions where they were inoculated in some media. Then these microbes which we have isolated, we went for their hormone analysis. We not only went for the abscisic acid, we also went for the hormone analysis for all the basic hormones in the plant. And we compared the hormone analysis of the microbe which had been obtained from the control region with the microbes which had been obtained from the stress region. And we studied the production of the important hormone like indole acetic acid, gibberellic acid, transient ribocyte from the stress and sites. And we targeted those strains which had the maximum production of the abscisic acid, and which is considered as the stress hormone. And these strains had some very good production of abscisic acid, and this work has also been published. Along with it, in the third part, what we do is that we combine these things. We combine the soil amendments and we combine the PGPR. We inoculate the plant and then we grow them under the induced stress condition, and we try to study the plant responses into these stresses. And we studied the effect of this inoculation, and we studied that these PGPR improve plant's root architecture, they improve plant growth, they improve plant physiology, osmolyte production, the plant hormone production, along with it, the antioxidant production. So these microbes, along with these microbes, we also had the soil amendments, and these organic amendments also, our results showed us that these organic amendments, they have increased the soil organic matter content, they have improved the soil aeration, the water infiltration capacity, and both the water and nutrient holding capacity, and most importantly, they even act as a fertilizer. They are providing energy to the roots, and also they are providing the energy to the soil microbes. Along with it, when we are talking about the soil amendment, one of the things that is very in or trend in nowadays, that is the nanobiotechnology. We have also tried to target the nanobiotechnology for the stress mechanism. And this was, our results show that these nanoparticles, they have the ability to tolerate the stress, particularly by enhancing the root hydraulic conductance and the water uptake in the plants. Along with it, they also show the differential abundance of protein. And more importantly, these nanoparticles are involved in ROS detoxification, the stress signaling, and the hormonal pathways. These are some of the results which we published. And these were very promising results, and they led us to some further studies. And then we went for the combination of the nanoparticles along with the biofertilizer. Biofertilizer here means where we have isolated the plant growth promoting isobacteria, and they were prepared a consortium, and we named them as a biofertilizer along with some carrier material. Later on, we combined them with some nanoparticles, and this was known as the nanobiofertilizer. Basically, this nanobiofertilizer, it consists of biofertilizer, which is encapsulated in the nanoparticles. And these nanobiofertilizer, they prove to be more cost effective, they are more potent, they are more efficient, and they were even considered as eco-friendly. And this is one of the review article which we published upon the improvement of plant responses by the nanobiofertilizer. Along with it, our results had been very promising for the nanobiofertilizer, and we have studied the synergistic effect of the plant growth promoting isobacteria with the silicon dioxide nanoparticles, and this was done for the amelioration of drought stress in wheat, and this work was published in Plant Physiology and Biochemistry. And which is these both are the very prestigious journal. The other work where we went a further step and we tried to prepare a compound that was named as nanosilica B. And this nanosilica B, it was also studied, it was investigated, and it was observed that it improved the activities of the metabolites in the triticum estima. And this work was published in the Plant and Soil. All of this, along with it, by the end, we got a product that was the nanosilica B or the nanobiofertilizer. And this nanobiofertilizer, which is our end product, this had been awarded in a competitive research program award by the Pakistan Science Foundation. And this is our project, which has been allotted for the same product. Here we have an industrial partner also, Ariga group of industries, which is a renowned for fertilizer company that is on board with us. And we are going to produce, inshallah ta'ala, this fertilizer for the field level analysis and also for the commercial scales. Then along with this nanobiofertilizer, we have also done some studies. We have also prepared another product, and this product is a bioorganic fertilizer. And this is where we have done some fermentation analysis. And this is also a very good product. We are working still upon this product. We are trying to go for its commercial production for the industrial collaboration and also for some large scale production through some project hunting. I hope we are going to have something relevant to this soon, inshallah ta'ala. These are some of the other publications, which these were rather preliminary publications, which led us all to those of our results. We did for QTL mapping at the different stages. We did for the multi-state variant. We tried to target the yield and yield components in the plant. Along with it, the effect of the stress hormones like abscisic acid, and also for the various amendments under the drought stress. We have a number of studies related to the isolation of these strains, particularly under the stress condition. Under the soil moisture is our main target in the drought-related studies. We have even tried to study the production of the exopolysaccharide. Exopolysaccharide is a kind of compound that is produced by the bacteria, which helps the bacteria to combat the stress condition under the natural conditions. So this exopolysaccharide production was also tested when we are producing the when we are isolating the bacteria, and this was their inoculation also proved to be very effective in ameliorating the drought stress in wheat. Along with it, we have also done a number of other studies, and we have done some characterization of the strains which have the ability to tolerate the stress conditions. These have been over a period of time. Similarly, the combined application of the strains and along with it, the soil amendments, they showed us very promising result for combating the stresses, particularly the drought stress, which we are talking about here. And we have studied in a number of crops, like we have studied them not only in the wheat, we have also targeted our research on the maize plant. We have also done some work on the canola, and we have also combined them with a number of other compounds like the PGPR compost and mineral fertilizer. We have tried to go for the maximum combinations to find out something that is practical and that is also applicable and performing in the field. This was the work which we had done for the drought, the basic stress which is Pakistan is facing. I hope you know that as the name of our university, that we are living in an arid agriculture university. We are all working over here. So aridity, the water shortage, the water less condition, they are more important for us as compared to the other studies. So this all which I had discussed by yet, this was related to the water stress. Along with the aridity or the water stress, there is another important stress that is limiting the plant production. I've not gone into much into the introduction of the soil tolerance or the things, because I assume that most of us have been in touch with these things, and we know all the things which how what is the level of these stresses in our country. In the second stress, we targeted for the salt stress. You know that the salt stress is even more damaging to the plant as compared to the drought stress, because in the salt stress, there is not only the physiological water shortage, but there also is ionic stress. So the plant adaptations are more complex, and these involve complex physiological rates, the complex metabolic pathways, and we have complex molecular or the gene networks. Plant adaptation to the salinity are of the three basic stresses. We have the osmotic stress, we have the sodium and chloride exclusion, and we also need to have the tolerance of the tissues, particularly for the accumulated sodium and the chloride. We did the same kind of the study over here, which we did for the wheat, for the drought. Here we did also for the QTL analysis. We tried to, first of all, to go for the morphological, physiological, and the antioxidants, which are performing an important role in the abiotic stress tolerance for the salinity. Then when we have defined these physiological parameters, then we went for the QTL. We tried to identify the position of these strains on particular chromosomes. And in this, we were able to determine the quantitative rate locus of mapping for the stress tolerance in the wheat and all for the 7 chromosomes. Along with it, when we targeted for the bacteria, which are the soil microbes or the PGPR, which have the ability to tolerate the salt stress, we also went for the production of the compatible solutes. I hope you are familiar with the idea of compatible solutes. These are the metabolic compounds. These are the metabolites. These are the osmoprotectants, osmoregulants. There are so many names for them. These compounds are basically being produced by the bacteria or by the plant, and they help in the water regulation. They help in the osmotic potential regulation, and in turn, they help in the water potential regulation in the plant. And in case of any kind of the drought, either it's the physical, that's the main drought, or the physiological, that's salinity, the production of this compatible solute, it is a determinant which helps us to differentiate between the tolerance and the susceptibility. So we isolated some of the strains from the salt stress region, and we checked the production of the important osmolytes like the proline, the reducing sugar, and total soluble sugar. We checked for the individual microbe, and we also went for the testing of the combination of the microbes under the stress conditions. And we studied the production under the various sodium chloride conditions like 0, 2%, 4%, 6%, 8%, and 10% NaCl. And based upon our result, we were able to find these are the microbes which were the best performing. And we were also able to determine and find out that these microbes even perform better when we have a group of microorganisms or the consortia. In the combination, these microorganisms are even performing better as compared to the individual effect. And this work has also been published. Then we also tested various soil amendments and PGPR inoculation under the salt stress conditions. And we observed that the biochar application, it increased the plant growth, the biomass, and the yield under the salt stress condition. It also helps in the increase of the photosynthesis, the nutrient uptake, and most importantly, the gas exchange parameters, particularly under the salt stress conditions. Along with the drought and the salinity stress was related to the ionic stress. So this ionic stress also led us some interest for the heavy metal stress. And because due to the increase in the number of the compounds which we are using in our daily life, increase in the pollution, we are having a large amount of the heavy metal stress in the plants. And these heavy metals are also causing toxicity, and they are this toxicity even includes the cellular at the level of a root, at the level of the cell, and there are various mechanisms by which, with the help of which, the plants they have the ability to combat these stresses for the individual metal ions. And most importantly, we have the sequestration of these metal ions, and along with this sequestration, we also have various detoxification mechanisms in the plant. These sequestration molecules they involve some chelators, some transporters, some detoxifiers. They all, particularly the plant vacuole, it is considered as a storage organ for all of these metal ions, and either these ions are sequestered into the vacuole or they are stored in some other non-productive part of the plant. But these stresses also cause the oxidative stress is very prominent in under the heavy metal stress. And we also observed that there was the production of the salt-related proteins, the hormones, and the antioxidant and the signaling molecules. And particularly, we also observed some deep shock-like proteins, particularly under the heavy metal stress. We try in the work which we have published, we have tried to study the effect of the assessment of the cadmium on the wheat in the hydroponic conditions. Along with it, we have also tried to study the effect of that. What if we have some water which is having that these heavy metals, and if it is being irrigated to some vegetables, how it can enter into the food chain and what are our risk levels. Along with it, we also try to isolate some plant growth promoting as a bacteria. Because these plant growth promoting bacteria, they are considered to be very effective in immobilization of heavy metal, and they also have the ability to reduce their concentration in the plant via precipitation, complex formation, and the adsorption. These are some of the strains which we have obtained: the Pseudomonas, the Bacillus. The strains are the Bacillus subtilis, the Bacillus cemensis, and the Pseudomonas fulva. We have done the whole genome sequencing, and these results have been very interesting, and we were able to locate some of the traits which were present in these bacteria, which can help the plant in combating the heavy metal stress. So we studied the effect of these microorganisms in alleviating the heavy metal stress, particularly like the chromium contaminated soil, or with the wheat, or in the where we had studied on effect on the brassica, and in combination with biochar also we studied the effect. And our research suggested that the biochar amendments, along with the PGPR, in combination with the metal tolerant PGPR, they are a very promising approach, and they can mitigate the soil contamination, particularly by the immobilizing the heavy metals and the organic pollutants. Another stress which we have done a great work in our lab, that is particularly upon the hydrocarbon contamination. We have tried to isolate some hydrocarbon degrading plant growth promoting as bacteria. We have also tried to produce some biochar for this hydrocarbon degradation, and we have characterized this biochar. And then we have evaluated the mitigation potential of PGPR and biochar for production of maize, particularly in the crude oil contamination, because maize is a crop which is very high energy demanding and it's very rapid crop. So this crop was very ideal candidate for the remediation studies. When we are talking about for this. So this biochar, it induced the polyaromatic hydrocarbons immobilization, and it helped us in the removal of the contaminants by the various methods such as ion exchange, volatilizations, and this PGPR produce a number of compounds post the biosurfactants, and which enhance the bioabsorption of these hydrocarbons and mineralize the hydrocarbons. And in this way, we were able to degrade the hydrocarbons from the heavy molecular weight to the low molecular weight compound. We have studied, this is a work which was published in the scientific report, where we have published the comparison of the plant growth promoting and remediation potential of biochar and the other thermal techniques for the oil contaminated soil. But luckily, we have been able to postulate a remedy, and this remediation of the crude oil contamination can be done by the biochar, which also enhances the plant growth. But most interesting result which we obtained was, the most interesting result which we obtained was biome, which we are able to create. And in this microbiome, we were able to have some biochar and PGPR engineering, and this all resulted in a system for enhanced hydrocarbon degradation. And this was a very promising strategy for environmental remediation, because when we are talking about the use of the PGPR and biochar in the bioremediation of hydrocarbon contaminated soil, and it's of some great importance practically and also internationally. This work was published in Environmental Pollution, which has an impact factor of above 9. And we are developing and applying these PGPR biochar-based remediating techniques. They can help us in mitigating the hazardous effect of these aromatic hydrocarbon contaminated soil. We are also improving some techniques of biopigmentation, the biostimulation, and we are using basically the PGPR and biochar in remediating the oil contaminated soils. So we have studied quickly concluding, we have studied a number of stresses. We have tried to target for the drought stress. We have tried to target for the salinity. We have tried to target for the heavy metal stress, and also some for the hydrocarbon degradation. But if we, as we were talking about the offshoot and the above ground and below ground targets, if we are talking about the shoot target, whenever we are talking about any stress, we have to go for the whole plant responses. And when we are talking about the whole response plant responses. The most important are the ROS, the reactive oxygen species. They are being produced under the stress. There are a number of them: superoxide radicals, the hydrogen peroxide radicals, singlet oxygen, and the scavenging of these ROS is very important. This involves both the oxidate with enzymatic and non-enzymatic antioxidant. And all of these ROS, if it is not scavenged, it can affect the membrane stability, which can affect the cell homeostasis. So along with it, whenever we are talking about all of this, the osmo protection or osmoregulation is equally important in the plant when we are talking about the whole plant responses. When we are talking about the above ground targets, the stomatal regulation is a very important. The gas exchange parameters are considered as a fundamental until we don't have any idea about what is happening in the photosynthesis. Is the main parameter which is governing in the plant. It's the main mechanism that is happening in the plant. The importance of the plant is even because of the photosynthesis. So we try to target upon the stomatal regulation and all the exchange parameters which are either directly studied or indirectly through the determining the effect on the photosynthetic pigment or determining the rate of the photosynthesis. The water conductions are very important. The xylem conductions, the water potential, the osmotic potentials are very important in most kind of the stresses. Even when we are talking about heavy metal stress, even it has the ability that it can hinder it, or even if we have some environmental pollutant like plastic or any other that is present in the soil, it hinders upon the soil water holding capacity, and it also causes the resistance in the soil moisture content and particularly the absorption of the water by the roots. Then we have some root targets also, and these root targets are most importantly, we have to make sure that the water is available in the root zone, because a plant only takes two things from the environment. One is the carbon dioxide and other is the water and nutrient. Water and nutrient are obtained from the soil. So until and unless plant does not get enough water, it is the growth is going to be stopped or inhibited. So wherever we are targeting, whatsoever we are using the PGPR or we are using the soil amendments, we target that all of them should maximize the root zone water availability, either by increasing the root area or by increasing the nutrient uptake, whatsoever. That's our main target. Along with it, the hormones are very important. If we have the higher level of ethylene is considered as growth retardant. So we target the ACCD minus producing microorganisms so that they should lower the ethylene and they should stop this growth restriction by the ethylene. The improved oxygen level, they are very important for the root growth, and due to this oxygen level, when they are increased, as a result, the growth of the plant is more. So these are the basic offshoot and below ground, the above ground and the below ground targets, which we have traced, which we have determined by yet, which can help in combating the various environmental stresses, which are supposed to be a danger in the future due to the climate change. On what's next, we are trying to go for the commercial production of the bioorganic fertilizer along with the silicon nanobiofertilizer. Silicon nanobiofertilizer is a bit ahead of bioorganic fertilizer. We will also try to have some work on the bioorganic fertilizer, particularly for its commercialization. So these are some of the awards and honor. I have received the first Professor Dr. Abdul Hamid Gold Medal for my work in the applied and environmental microbiology. I have also received a research award from the university and some research publication award. And I'm also happy to share with you that I'm the president of Pakistan chapter of Asian PGPR Society. And there is a conference of this Asian PGPR upcoming in August this month. So if anyone of you is interested in plant promoting bacteria, you can join them. So this is from where I started. I did my PhD from Dr. Asghari Bano from Kaiser University with some of the friends which are still with me. And then I got a very good and I joined a very good department, and we have some great colleagues with us and a very conducive kind of environment which helped us and my lab and my student in achieving all we had done by that. And these are the students whatsoever. A supervisor is nothing without the students. These are my PhD students and along with some MPhil students, and which have been playing a very important role in all of these studies and performing all the work and also doing the write-ups. And in the end, I'd like to thank you. And if you have any question, I'll be happy to answer. If anyone has any question, I'll be happy to answer.
Hello,
Yes, sir.
you please go ahead, please.
This I have finished my talk. If anyone has any questions, I will be happy to answer.
Hello,
Yes, sir, we can hear you.
madam, can you please hear me?
Yes, sir, we can hear you.
Okay, so house is now open for question and answer session. If anybody has question. Okay, if there is no question, I think Dr. Noshin, thank you very much for delivering a very informative talk on the subject. I have seen some of your achievements, which I think I appreciate very much. And I think it was very informative talk. And at the end of the talk, I would like to thank all the participants for their participation. And thank you again.
Thank you, sir.
Allah Hafiz. Allah Hafiz.
Okay.